Last week, I gave you a pretty broad overview of how a trait is passed down from parents to offspring. This week, we're going to get into more specifics of how that actually works. So, by the end of this lesson, you're going to be able to show how alleles are inherited and how they influence traits. To begin with, we've got to go back to DNA. So, you'll remember that DNA is found inside the nucleus, it's found in segments, and those segments can condense into these tightly coiled structures that we call chromosomes. You, a human, have 23 pairs of these chromosomes, so 46 of them total in your body, in every single one of your cells. 22 of those pairs are what we call autosomes, meaning they're identical to each other and they're something other than the unique sex chromosomes. The prefix auto there meaning self or same. The last pair, they're the sex chromosomes, and we're going to talk about them a lot more later on. So keep that information at the back of your brain. You will need it later. If we zoom in a little on these chromosomes, you'll remember that after replication, which happens in kind of the middle of the cell's life cycle, each of those chromosomes, those X-shaped structures, are made up of two sister chromatids, which have perfectly or nearly perfectly identical DNA. So for those of you keeping score, that's 4 total copies of each segment of DNA, or 4 total copies of each gene, because each strand of DNA contains genes that code for proteins. Interestingly, we've figured out that the sequence of about 90% of the human genome, and it turns out that only about 4% of that DNA actually codes for proteins. The other 96% that we've worked out so far is what we call junk DNA, and we really don't know what it's doing there or why we've got it. But in the pictures on your screen, you can see some of the areas that we have mapped on chromosomes and some of the proteins that we've identified that those chromosomes produce. So, for example, chromosome 1, which is the largest of the 3 you see there, the one on the left, has genes that we think are involved in Alzheimer's disease and prostate cancer and glaucoma. Chromosome 11, which is the upper one, this guy right here, has some genes that we think code for diabetes. And the third one there, the smallest one, chromosome 17, has genes that are involved in breast cancer and in tumor suppression. Now, notice I said before that you've got 4 total copies of each strand of DNA, but those 4 copies aren't all identical to each other. You've got an identical pair in each chromosome, but the 2 chromosomes aren't necessarily identical to each other. Remember, half of any organism's DNA comes from each one of its parents if we're part of a species that's doing sexual reproduction. So if we pretend for a minute that this image was showing my chromosomes, which it's not, but it could be, for chromosome 1 up there, one of those would be the genetic material that I inherited from my father, and one of those would be the genetic material that I inherited from my mother. They'd have the same genes in the same locations as each other, but the exact nucleotides that make up each gene could be different, possibly very different. In fact, considering how different my parents look from each other, there are probably some locations where they are very different. Now, I mentioned our dear friend Gregor Mendel last week. Well, he came up with a law that explains how this works. We call it the law of segregation, and it says this. First, a gene can exist in more than one form. We call these different forms of genes alleles. My example of this from last week was the LCT gene, which codes for the lactase protein, which comes in two different forms, two different alleles. Next, an organism will inherit exactly two alleles for each trait, one from each of its biological parents. This happens because of the third point, when gametes, or sperm and egg, form during meiosis. Each of those gametes will randomly receive an allele for every trait, for every gene that it has. Finally, the ultimate trait that an organism will have depends on which alleles it gets. When there are multiple alleles, some of those alleles will be dominant over others. So the dominant trait will always be expressed. A recessive trait would only be expressed if the dominant trait isn't around. So, as a reminder, we talked about this last week too with the LCT gene. The gene for normal lactase production is dominant. So if you have at least one copy of that gene, you can digest lactose. If you get two copies of the recessive gene, you produce much less lactase than normal, and you cannot digest lactose. Here's a reminder and some more in-depth information about how Mendel figured all of this out. Remember, Mendel did his work with pea plants. So let's look at flower color in pea plants. And pea plants are very useful as models for figuring out how genetic works, it turns out, because they have some very easily identifiable traits. And because they can both self-pollinate, which means one plant by itself can produce offspring, and they can cross-pollinate. So you can breed two plants together to produce offspring. So you can very easily control what two parents you want to get offspring from. When Mendel set up his experiments, the first thing that he did was to breed up lines of his pea plants that always bred true for certain traits. So in this example, a purple-flowered plant that, if it self-pollinated, would always produce purple-flowered offspring, and a white-flowered plant that always produced white-flowered offspring. This was his pea, or parent generation. So he took plants from that pea generation, one with purple flowers and one with white flowers, and crossbred them. And this created his F1, or his first offspring generation. And all of those F1 plants came out with purple flowers. Now, Mendel continued his breeding program by self-pollinating those F1 plants to make an F2 generation. And this is where he saw something really interesting, because those all-purple F1 plants produced a pretty near to perfect ratio. 3/4 of their offspring would also have purple flowers, but 1/4 of those offspring had white flowers. This precise mathematical relationship happened no matter which of his target traits he was testing. And it was the key that allowed him to work out exactly what was going on. All right, so in order to understand what's going on with Mendelian genetics, we use a tool called a Punnett square. I think I showed you one of these last week, but just in case you missed it, it essentially looks like a 2x2 table in its simplest form. In a Punnett square, we can plot out the alleles that each parent has and work out from there what the possibilities for the offspring are and how likely each of those possibilities is to actually happen. So let's get some parents in here. And we'll start with Mendel's pea generation. So we've got on the left one purebred purple plant, and on the top one purebred white plant. Because we know that these plants always breed true, we know the genetics for each one of these plants. We know that the purple plant has to have two copies of the allele that codes for purple flowers, and we know that the white plant has to have two copies of the allele that codes for white flowers. Now, I've picked the letters representing those two ideas that you see here for a reason. I chose capital F for this gene because it's the first letter in flower. So, highly complex reasoning here. But notice that I gave capital letters to the purple plant and lowercase letters to the white plant. This is because I know from Mendel's experiments that the purple flower gene is dominant and the white flower gene is recessive. As a convention, we always give the dominant gene a capital letter and the recessive gene a lowercase letter. Just as a note, here's a couple new vocabulary words. These letters that represent what alleles the plant has are also known as the plant's genotype. Remember, genotype refers to the genes. And the result, what trait that genotype produces, is called a phenotype. So in these plants, the phenotype that we're looking at is what color their flowers are. The capital F genotype produces the purple flower phenotype, and the lowercase f genotype produces the white flower phenotype. Now, take a look at that purple plant. When it produces gametes, those gametes will randomly get one copy of its purple flower alleles. So from the purple plant, we know that they have to get a capital F allele. Similar concept for the white plant. It only has the lowercase f allele, so that's all it has to pass on. I arranged those on the outside of my Punnett square. On the inside, in each of those squares, I'm going to say, okay, so what if the offspring gets this allele from one parent and this allele from the other? And I fill that into my table. So in the first square, we have an F allele in that row, capital F allele from the purple parent, and a lowercase f allele in the column from the white parent. So we know that the offspring is going to have one capital F allele and one lowercase f allele, making its genotype capital F lowercase f. Since purple is the dominant gene, we know that that plant is going to be purple when it comes to its phenotype. In fact, let's just fill this in for all of this Punnett square, because all of our F1 offspring are, as it turns out, going to have the same genotype. They all have to get the capital F allele from one parent and the lowercase f allele from the other parent, because that's all that they've got. So just like Mendel observed, the F1 generation will be entirely made up of purple flowered pea plants. But these purple flowered pea plants are different from their purebred purple flowered parent. Say that 5 times fast. So continuing to say this true breeding purple flowered plant and the like is going to get old quick, and I have already messed it up so many times. So it's time for some new vocabulary. Those parent plants, we call them homozygous, meaning they have 2 copies of the same allele. The purple parent is homozygous for the dominant gene, or homozygous dominant for short, and the white parent is homozygous recessive, meaning it has 2 copies of the recessive allele. All of the F1 generation children here are heterozygous, meaning they have 2 different alleles. Same phenotype as the purple parent, they all have purple flowers, but a different genotype. Here's where the math comes in, because if we self-pollinate those F1 plants, we get the F2 generation. And now there's variety, because when a plant with a genotype capital F, lowercase f, breeds, instead of having a 100% chance of passing on the same allele like our last generation had, it's got a 50-50 shot now. Half the time it's going to pass on the capital F allele, and half the time it's going to pass on the lowercase f allele. What that means in terms of our results is 75% of the time, or 3 out of 4 of our plants, at least one dominant allele gets passed on. But 25% of the time, both gametes have that lowercase f allele. And what gets produced is a white flowered pea plant. Exactly what Mendel observed and what allowed him to solidify his theory. Now, remember, Mendel was working with 7 different traits in pea plants. He wasn't just studying the color of the flowers. He was studying the size of the plants and the color of the pea pod and the color of the pea inside the pod and the shape of the pea inside the pod and a couple of other traits that I'm not going to remember off the top of my head. So, what happens when you're tracking how multiple genes are passed down? Well, Mendel had a law for this too by the end of his research, the law of independent assortment. And what this says is that traits are inherited independently of one another. So the color of flowers that a pea plant produces will not impact what color the pods will be, whether it makes yellow peas or green peas. Each of those traits is inherited completely independently of the others. So tracking that genetics could look something like this. You can make a Punnett square for multiple traits. It just means a little more work. So here we're looking at the shape of the pea a plant produces and the color. They can either produce round peas, which is the dominant trait, or wrinkled peas, which is recessive. Similarly, they can either produce green peas or yellow peas, yellow being dominant and green being recessive. So in this diagram, we've got capital R being round peas and lowercase r being wrinkled peas, and capital Y for yellow peas and lowercase y for green peas. Now, we could have gone with S for shape and C for color, but here's my other rule for picking letters. It is so much easier when you choose a letter that you can easily tell the difference between upper and lowercase. In this example, over on the left, they've crossed 2 true breeding homozygous plants to get an F1 plant that is heterozygous for both traits. So these plants, these F1 plants, have a 50-50 shot of passing on the dominant or the recessive gene for both traits. We set up a table again, but this time each row and each column have to represent one unique possibility for what combination of traits each parent could pass down. So it could pass on the capital R allele with either capital Y or lowercase y, or it could pass on the lowercase r allele with either capital Y or lowercase y, which results in a Punnett square that looks like this one. So there were 4 options for combinations of traits that could be passed on. So each side of the Punnett square has 4 columns or 4 rows. We call this kind of Punnett square, where you're looking at 2 traits, a dihybrid cross, di meaning 2. And you can make them more complex than this. We could have added flower color back in here and made a trihybrid cross, where we looked at 3 traits or beyond. Doesn't that sound like fun? As a high school student, I had to create and turn in several trihybrid crosses, and I think we even did like the next level up where we did 4 traits, which I'm not even sure that there's a proper term for. And I actually found this to be quite soothing, but I realized that it is not going to be everyone's cup of tea. So for the authentic Miss Smith in a high school experience, that is an option this week if you really want to make some of these Punnett squares of your own. But I am not going to force you into it. There are other options if this all sounds incredibly boring to you. Now, we've been talking about Mendel and his research, and it's important to note that all of this was taking place in the mid-1800s, back when we still weren't really even sure how traits were passed down from parents to offspring. Like, this is just a few years after a guy named Lamarck suggested that if an animal acquires a trait, like if a giraffe spends its whole life stretching to reach fruit on high branches and its neck grows longer as a result, it could pass that acquired trait on to its offspring. Or like if a starfish loses a limb, it could create 4-limbed baby starfish. And this was right around the time when Darwin was just starting to come up with his alternative model to that theory. So we had very few clues about how heredity worked at the time, which kind of begs the question, how much of Mendel's explanation of genetics is actually right? And surprisingly, a lot. Mendel was incredibly lucky, and he pretty much got it down with, a couple of exceptions. See, not all of genetics behaves according to Mendel's lovely simple laws. When a trait is controlled by a single gene, and when that gene has two alleles, it is going to follow the rules that I have just given you for Mendelian genetics pretty much perfectly. And when you've got multiple genes, under the right circumstances, they will also follow Mendel's rules pretty much perfectly. But we're going to look at some exceptions. And first on the list is other patterns of expression, because it turns out that Mendel's idea of dominant and recessive genes wasn't the whole picture. There were a couple of other options. For example, two alleles can show incomplete dominance. Imagine our pea plant again. If we had bred that purple flowered plant to the white flowered plant, and the offspring had been plants that produced flowers that were sort of in between the colors of the two parents, that would show incomplete dominance. Neither allele is perfectly dominant over the other, so both versions of the color protein are produced, and the phenotype is some blend between the phenotype of the two parents. Pea plants don't do this, but a lot of other flowers do. It's very common that if you breed a red flower to a white flower, the offspring will be pink. Or you can have another option, codominance. This is where, when you've got two dominant alleles, both of them get expressed, and you see one show up in one place and one show up in another place. So in plants, you tend to see this looking like some sort of a speckling pattern, like you see in the flower here, where some parts are white and some are the dark pink color. That flower would likely have had one white flowering parent and one dark pink flowering parent. This is also part of how human blood typing works, but human blood is even more fun, so let's go talk about that for a second. When we talk about blood type, we're talking about what kind of surface proteins your blood cells have and what kind of antibodies you produce to fight off foreign invaders. See, your body is all about keeping the outside out. And it's very good at hunting down and eliminating outside threats. Sometimes too good, which is how you end up with all kinds of autoimmune disorders. For example, I have type B blood. In humans, there are 4 possible blood types that are the result of 3 possible alleles for human blood cell proteins. So while I can only inherit 2 alleles, there are 3 total that are out there in the gene pool, which complicates inheritance a little. So I inherited 2 alleles, and because of my parents' blood types, I can actually tell you exactly what my genotype for blood surface proteins is. It's BO. Yes, laugh, go ahead. But what that means is that I inherited from my mother a copy of the B antigen allele. My mother also has type B blood. So I produce the B shape of surface proteins in my blood. From my father, I inherited the O antigen allele. That allele doesn't produce any surface proteins at all. So my blood group is B because I only produce B surface proteins. B is dominant to O. The only way to get a blood cell with no surface proteins is if you make no protein at all because you have 2 copies of the O allele. But my father's blood group is A. So I had a 50-50 shot of inheriting a B allele from my mother and an A allele from my father. A and B are codominant, just like in the flower that you saw before. So if someone inherits an A allele from one parent and a B allele from the other parent, they will produce both surface proteins, so resulting in an AB blood type. Blood group inheritance is fun. And it gets even more complicated because it turns out that not all traits, in fact, not even most traits, are Mendelian because they're coded for by many genes, not just one. We've identified 3 genes, for example, that play into what color your skin will be. And there could be more that we just haven't identified yet. And skin color is even more complicated because it's not a you're either purple or you're white kind of trait. There aren't just 2 options. There's a whole range, what we call continuous variation. Even within our human attempts to classify people based on what color their skin is, if you say that somebody is white or that they're black, that doesn't really give you a good picture of exactly what their skin color is going to be. If I picture in my head somebody with black skin and I tell all of you to do the same thing, we will likely come up with, let's see, I've got roughly 40 living systems students. So, roughly 40 different images of what that skin color is. And you certainly can't use the information that I have white skin to predict what my genetic makeup looks like because there are so many different options that produce this color. And as you can see in that diagram, 2 parents, even if they've got very similar skin color, could produce offspring with skin color that looks very different just because of the random nature of which alleles go into what gametes. So if those 2 people in that diagram have a child, even though they've both got a pretty middling skin tone, that kid has the same chance of coming out with either much darker skin or much fairer skin than its parents have. Moving right along, there's also that pesky law of independent assortment. You know, the one where genes are totally random and what allele you get for the flower color trait doesn't impact what allele you get for the color of the pea pod. But in the real world, we know it doesn't work that way all the time. If it did, it would be statistically impossible for me to sit here in front of you today and tell you that an estimated 80% of redheads have freckles. It turns out that genes that are on different chromosomes from each other follow that law of independent assortment. But when 2 genes are on the same chromosome, the closer they are together, the more likely they are to get passed down together. So if one of your parents had alleles that produce red hair and freckles, they were likely to pass those down to you as a package deal. This has to do with crossing over. Remember, during the early part of meiosis, the chromosomes that you got from each of your parents can swap chunks of their DNA. So you could end up in one gamete passing down your mom's nose shape and the size of your dad's ears. But the closer 2 genes are to each other on the same chromosome, the more likely that they are to end up on the same chromosome, even if they are swapped. So you get the red hair allele, odds are pretty good that you're going to get the freckles allele to go with it. This is where I say that Mendel got incredibly lucky because by sheer dumb luck, he picked a bunch of traits in pea plants to study that were on different chromosomes. He would not have gotten the results that he got if he had picked traits that were on the same chromosomes. Finally, we've got to go back to that idea of autosomes and sex chromosomes that we talked about earlier. I mentioned that autosomes are identical, meaning the genes that I've got on the copy of chromosome 1 that I inherited from my mother are the same genes that I got on chromosome 1 that I inherited from my father. Maybe not the same alleles, but the genes, the proteins, are the same. For the sex chromosomes, that is not true. The X chromosome and the Y chromosome have very different DNA on them. The kinds of changes that your body will go through during puberty are coded on the X chromosome along with some other miscellaneous proteins. All the Y chromosome is an override switch, basically, that says, no, don't make those changes, make these changes instead. Which means that everything else that's on the X chromosome, if you're a person with one X and one Y, well, you've only got one copy of it. So, this is the reason that certain genetic diseases like colorblindness and hemophilia are much more common in people with one X and one Y chromosome than in people with two X chromosomes. If you inherit two X's, you've got a backup copy. If one of them doesn't work, maybe the other one does. But if you've got one X and one Y, and that X has the disease causing allele, you've got nothing to compensate for it on the Y chromosome, and you are going to have that genetic disorder. As an aside, I hope you're noticing some of the language I'm using here because, yes, these two chromosomes are involved in determining your biological sex, but it is vastly more complicated than if you get two X's, you're female, and if you get XY, you're male. There is a supplemental video this week dealing exclusively with this topic. So if you're interested, you can choose that for your gain knowledge this week. But meanwhile, we are going to go on back to the subject of genetic disorders and back to how traits are passed down in general. It can be incredibly useful to be able to track how traits are passed down through generations. So if you don't want to make Punnett squares this week, you're going to be focused on how we track traits through generations using a tool called a pedigree. This is a pedigree chart. And what you're seeing here is the way that a particular trait in this chart, it is the trait of having a white forelock in your hair, kind of like the one that I've got in my hair here. So on this chart, you're going to notice some features. First of all, there are circles and squares. A circle represents a female member of this family, and a square represents a male member of the family. You will notice that there are blue squares and white squares on this one. Sometimes you will see them in black and white where they're just filled versus empty squares. Generally, the filled in square indicates that a particular trait is present, and an empty square means that a particular trait is absent. Some pedigrees will use a half-filled shape to represent someone who is a carrier for a gene but does not express it. This one doesn't, but the ones that you will see if you choose the pedigree assignment will. And then the lines are showing relationships. So a horizontal line represents that these two people are a couple, that they have produced offspring together. And then a vertical line and a bracket coming down from it, that shows the offspring of that particular relationship. So take a look here in that red circle. Notice that you have two parents with the white forelock gene, but they've produced a child with no forelock. Now, the white forelock gene in this particular example is dominant. So that means that no white forelock is recessive in this example. Because the no white forelock gene is recessive, we know that this white circle child must have a genotype of lowercase f, if I pick F to represent my forelock gene. From that, we can predict the parents have to be heterozygotes. They have to have capital F, lowercase f as their genotype, so that each one is carrying but not expressing the recessive gene. We could go through the whole family tree this way. And for at least a few of the family members, we could predict what their genotype must be based on what their phenotype is and what phenotypes their children and parents have. We can also use a pedigree like this to figure out whether a trait is dominant or recessive. See, if the white forelock gene was recessive with our little family unit in the red circle, those two parents, if the white forelock gene was recessive, would only be able to produce offspring that have the white forelock. Because if there was any copy of the no forelock gene in there at all, and that gene was dominant, it would take over. One of the parents would have the forelock. Make sense? Then it's time to go try it out yourself. Go do some work either with the pedigrees or with the Punnett squares. Go do some more learning on how traits are passed through generations and have fun. Be good, be safe, be kind. Don't do anything I wouldn't do. Wash your hands. And next week, we'll wrap some of this stuff up.